Good morning, everyone. Welcome to today's Cannabis and Hemp Research Forum hosted by the UC Davis Cannabis and Hemp Research Center. I am co-director Li Tian, along with my co-director, Professor Cam Carter. The purpose of our monthly forum series is to inform our campus community, members of the cannabis and hemp industry, and the general public on cannabis and hemp research being done at UC Davis, and also to provide an opportunity for guest external researchers to share their expertise, such as our invited speaker for today's forum. As the presentation is underway, you will be able to pose written questions using the Zoom Q&A feature. Our presenter will save some time at the end to address your questions. This presentation is being recorded, and the recording will be made available in the next few days on the Cannabis and Hemp Research Center website at cannabis.ucdavis.edu under the Education tab. Now, I'm very delighted to introduce today's forum speaker, Dr. Mark Lang. Dr. Lang is the interim director and professor of the Institute of Biological Chemistry at Washington State University. Dr. Lang is also the chief scientific officer of Dewey Scientific. He is a renowned expert on terpene metabolism. For today's forum presentation, Dr. Lang will discuss their findings on terpene synthesis in cannabis. Welcome, Dr. Lang. Thank you for taking the time to present your research.
Well, thank you, Lee, for the very nice introduction and also for the invitation to join. And good morning, everyone. I will be talking about terpenes, and I presume that you've heard about the topic in previous presentations. So I will keep my introduction relatively short, but I will try to cover a few very important aspects of terpenes and terpene metabolism. One of the things that we've been interested for many, many years is specialized cell types, tissues, and also anatomical structures that contain these specialized cell types. And one of those structures are glandular trichomes. And those are modified hairs that you find across the plant kingdom, and oftentimes they're used to store larger amounts of specialized metabolites. And in cannabis, the relevant metabolites would be the cannabinoids and terpenoids that are accumulated primarily in the glandular trichomes. You've probably seen pictures like this. So these whitish structures that you see there that kind of look like hairs, they are actually, for the most part, modified hairs that synthesize and then store primarily cannabinoids and terpenoids, but also some other specialized products. What's kind of interesting in this context is that both the cannabinoids and terpenoids have a common biosynthetic origin. So I've redrawn the pathway to a certain extent. And down here, we have a precursor for cannabinoids. And what I've done with the color code is to show where the different parts of the molecule originate from. So the core resorcinol structure comes from malonyl-CoA, which is a precursor for fatty acid biosynthesis. There are polyketide synthase type enzymes that fuse C2 units from the malonyl-CoA and also add a side chain down here, which is shown in blue. The side chain in this case coming from fatty acid breakdown, and hexanol-CoA is the precursor for the most commonly accumulated cannabinoids. So you have a resorcinol structure with a side chain on the one hand, and then you're adding a moiety that comes from terpene biosynthesis. In most cases, it's a carbon-10 molecule, but it can also be carbon-15 and possibly others. So as I mentioned, we have three different pathways that contribute to the formation of the cannabinoids. And from the geranyl diphosphate and also other precursors that come from that same pathway, we can generate terpenes, various types of terpenes. So in a way, the biosynthetic scheme already shows you that there is a link between the terpenes and the cannabinoids. However, that still had to be established by using various genetic and biochemical approaches. So how do you connect genotype with chemotype? I'm going to show you some data that we published a few years ago. The main person involved in the work was Jordan Zager, who was a graduate student in my lab and then later became the CEO of Dewey Scientific company that I co-founded. Iris has done most of the work on cloning genes, putting them into expression vectors and helping us on the molecular biology side. And then Shri was instrumental for the identification of the functions of terpene synthase as part of this project. So what we did is we profiled terpenes and cannabinoids in 9 samples that we collected from Shadowback's farms in Southern Oregon, where we have had access back in the mid to late 2010s. We then isolated glandular trichomes from there. So we developed a protocol that allows you to do this and generated transcriptome data using the RNA that was isolated from those glandular trichomes. So the key here is that when you're working with RNA that is expressed specifically in these specialized structures, what you do is you access the specialization. You make the best use of the specialization of these cell types. So oftentimes you will find that there's only a couple of thousand different types of transcripts that are expressed in those cell types in comparison to possibly tens of thousands in regular photosynthetic cells. So with the specialization then comes an enrichment in the transcripts that might be of interest for the pathways that you're trying to elucidate. Then we differentiated samples based on statistical analyses, and most of you have probably heard of those. Those are multivariate statistical analyses, including principal component analysis, or PCA, hierarchical clustering, and others. And those are commonly used to differentiate the overall profile of whatever your analytes are in complex comparisons between samples. So we started doing this with metabolites. So we had the 9 different chemovars that are listed up here. And what we're asking here is if we take all of the chemicals that we profiled and we look for patterns in the accumulation of these, can we differentiate them with statistical analyses? So on the left-hand side is one of these PCA, principal component plots. And you can see that the replicates cluster very closely together in a 3-dimensional space, whereas the various different sample types actually separate out very nicely. The principal components themselves are not anything that is directly meaningful. However, you can extract meaning from them if you look at what's called the component loading. So you can actually look at which metabolites contributed the most to the separation of the various sample types. I won't go into the details there, but it gives you some idea of the power of these types of approaches. Now, we did a slightly different approach to separate out samples on the right-hand side. And again, it's a statistical analysis that groups the sample types based on the overall metabolite profiles. And in this case, they actually separated out into what would traditionally be called a sativa or an indica type. Now, this nomenclature is outdated. In most cases, this will not work anymore because this was implemented at a time when we didn't really understand what truly differentiates the different chemovars. But in this particular analysis, it actually separated them out, just to make that point. then we did the exact same analysis with the transcriptome data. So again, you're looking at the entire data set and you're asking, can we differentiate the different sample types based on the overall expression patterns? On the left-hand side is a principal component plot, and you can see that again, the replicates cluster very closely, but the sample types separate. The same is true for a hierarchical clustering analysis, where you again separate out the sample types. And where this dotted line is, that again separates the sativa from the indica types. So just with the caveat that that is not the best characterization of those, but in this particular analysis, we were actually able to differentiate them. We then used an approach called network analysis that allowed us to look at both data types. So what we're asking now is which of the genes that are expressed across the various different chemovars correlate with specific chemicals that are accumulated in the glandular trichomes where these genes are actively expressed. And we had a total of 8, I believe it was, different clusters. And when you look at the cluster that is shown in yellow down here, and blow this up, you can see that the different chemovars that are given different colors on the scale here did the gene expression within each of those did to a certain extent correlate with the accumulation of certain types of terpenes. And on the right-hand side, we have a number of cannabinoids. Okay, so the amounts of these terpenes in some of those chemovars correlated directly with the patterns of gene expression for certain types of genes. And this is then shown in panel C down here with a little more resolution. So here you can see which of the genes that correlated with certain types of chemicals were expressed most similarly across all the different chemovars. And where you see the nomenclature added up here, CS, TPS14, for example, and others, these are terpene synthases that we later characterize functionally. I will not talk about this in the present talk, but it just gives you some idea that pretty much all of the genes that we characterize were very closely related in terms of expression patterns. And if you take what you see on panel C here and map the genes onto a pathway scheme, then it becomes obvious that the genes that are required to generate the proteins that are necessary to take up some kind of carbon source, glucose, raffinose, sucrose, whatever it might be, and turn it into end products such as sesquiterpenes, monoterpenes, and the cannabinoids. You can see that the genes that are involved in those pathways are all apparently co-expressed based on our analysis. And this is true across the various different chemovars and was a very powerful tool then for us to identify genes for further follow-up analysis. A point I was going to make is that the diversity of cannabinoids, as it is known currently, far exceeds what we typically analyze. So with Jordan together, wrote a review that was published a couple of years ago. We looked at all the cannabinoid structures that had been characterized thus far and counted roughly 130 unique structures. But if you think about how many are analyzed, it says 20 here. Oftentimes it's less than that. Many of the routine analyses actually focus on only 10. Now, for some people that might be sufficient because they're only interested in potency, which is primarily related to the amounts of tetrahydrocannabinol or THC accumulated. But if you're interested in a broader picture, then obviously you're not capturing the true diversity that is known in the cannabinoids. And that is illustrated again here in this overview scheme. So I'm just showing the core structures that are known. We start with cannabigerolic acid, or CBGA, which is the precursor, and then it branches out into various different classes. And then because there was not enough space, I added what is on the lower part of that figure in the paper. So again, it starts with CBGA and then leads into various other products. And as I mentioned, only very few of these classes are captured by routine analyses. Within these classes, it's only just a couple or 3 or 4 cannabinoids that would be covered by routine analyses. So obviously a shortcoming. The same is true for the terpenoids. If you look at library that is sold for the analysis of GCMS data that was initially generated by a person called with the last name of Adams. There are roughly 2,200 structures in that library. And again, for most of the cannabinoid and terpene analyses that I've seen for cannabis, only 20 of those are routinely analyzed. So again, we're really not capturing what is possible based on the technology. We're really only capturing those that are most common. And in some cases, that might be a mistake because we're actually missing something. So one of the key questions then is, what are those terpenes that really matter in cannabis? And I wanted to make a few comments about how you approach that issue. Now, for most people, you will be purchasing a standard mix from a commercial source, and that will be your first step in establishing a library. And then when you're running GCMS or other types of analyses, you will compare your retention times potentially, or maybe just your mass spectra with those of those authentic standards that are in that standard mix that you just purchased. So I asked Josh Polito, who is a graduate student in my lab, to investigate this a little bit. We looked at the commercial standard mixes that you can get from various different sources. And what's quite frustrating for us is that oftentimes there are things in there like menthol or pulegone. Now, those do not occur in cannabis. They are typical terpenes found in mint essential oils, but not in cannabis. So why would you put those into a cannabis mix? What it does is that if someone runs a chromatogram and they get a peak, and this peak, in terms of its retention time, aligns with menthol, for example, then you might assume, well, I may have menthol in my sample, so this is what I'm going to report. Now, when menthol is not in there, and it's just by coincidence that you have something that is somewhat similar and gives you a peak at the same retention time, then that's a real problem. The other thing that these mixes do is that they oftentimes list stereochemistry, like R plus limonene. Now, this is a molecule that accumulates to high levels in citrus peel, but not in cannabis. So again, oftentimes you are misled by what's in this mix, and then people report these kinds of compounds, and then you're creating an entire literature that is problematic because of multiple misidentifications of peaks. So just to make that point, we ran extracts from various cannabis chemovars on a chiral column. So chiral columns allow you to truly separate enantiomers. So this question about minus limonene and plus limonene could actually be investigated. Now, what we found is that in any chemovar that accumulates limonene at fairly high levels, you will find that it is only the minus limonene that accumulates. The plus limonene is a tiny little peak, and that is just because the enzyme that generates the limonene is not 100% specific for generating just one enantiomer. Now, there are chemovars where you find slightly enhanced levels of the plus enantiomer, like this one here, E14. But this is in a background where you have a different chemical, in this case it is terpinolene, that is dominant. Okay, so limonene is only a very small part of the overall terpene profile. And yes, you can sometimes have higher levels of plus than minus limonene, but it is an exception. For the most part, you find the minus enantiomer in cannabis. So reporting on the plus enantiomer just because that's in your list of chemicals that you get from chemical suppliers is a real problem. And this is just more evidence that it's mostly the minus enantiomer that accumulates. Then another aspect of the analysis is that you have various different options of processing your samples, and then also analyzing your samples in various different ways. And each one has its challenges and advantages. So again, I asked Josh to look into this and run analyses using a range of different processing methods. So if you think about a sample, let's say you just have a flower sample, you could decide to just analyze this whole, okay? So you can take your flower sample and you can put it into a vial and you could process it that way. Now, the other option that you have is that you could generate a homogenate. The homogenate means that you might throw it into liquid nitrogen and then grind it up with a mortar and pestle, for example. So in that case, what you're doing is you're destroying the natural structure of your sample and you're turning it into a more homogeneous mixture. And as you might imagine, there could be differences between those two processing methods. Now, then you could decide to take the headspace above your sample to analyze terpenes. So the terpenes that we're interested in are all volatiles. So they will be released from your sample. Okay, there will still be some in there, but even if it's intact, a lot of the terpenes are going to be released. So this air space above the sample is called the headspace. So you could then take a fairly large volume syringe. You can take up the headspace and inject it onto a gas chromatograph, for example, for separation of the terpenes and then downstream analysis. That is one valid method. Alternatively, there's a method that a lot of people have used for samples where the volatile emission is not very prominent. So if you're interested in also analyzing constituents of lower concentration, what you might do is that you might insert a solid phase microextraction tool into the vial that contains your sample. Okay, so what happens in this case is that you have a material that coats this needle here that absorbs terpenes. So essentially, you're concentrating the terpenes on this fiber, and the fiber then is moved over to the injection port of GC, and you inject. So that's a slightly different technology compared to analyzing the headspace directly. Now then, you could also decide that you generate a homogenate. You could then add a solvent to extract terpenes into. You could spin this down, and then take the supernatant that contains the dissolved terpenes, and then make a liquid injection. Okay, and you could imagine, as shown here on the left-hand side, that you do this in a combinatorial fashion, where you're changing the sample processing, the way you accumulate the terpenes, and also how you collect them. Now then, I will not go through the differences in chromatograms that we'll get with different liners, but what is important to note here is that you have different choices of columns. And in those cases, the separation of terpenes might be substantially different. The HB5MS is a column that is multi-purpose. It is used for a lot of different analytic challenges, and it will separate terpenes reasonably well. Now, the DB wax column is something that was developed particularly for terpene analysis, and in the monoterpene region, it gives you a far better separation than this HB5MS column. So you might say, you know what, I actually want to use that DB wax column because it gives me better separation. And then, as I mentioned before, there are chiral columns, and in this case, we tested the cyclodex B column, and that will give you a separation of the enantiomers. Now, why is this important? Enantiomers look very, very similar. There's only one carbon where the four-dimensional arrangement of residues is different. However, our nose can differentiate, okay? One of the typical examples I like to give is that the minus carbon smells like spearmint and plus carbon smells like caraway. So it's very, very different, although it's essentially the same molecule. And that is true for some of the terpenes that are accumulated in cannabis as well. So if you're interested in modulating aroma, you have to understand the stereochemistry. And then at the back end, you have choices of different detectors. What's very commonly used is a mass selective detector, or MSD, where you would be detecting, or you would be fragmenting molecules that come into the ion source, and the patterns of fragmentation that you get are characteristic of a certain compound type. So you can use that particularly in comparison to what you have in spectral databases to identify peaks. Another choice might be a flame ionization detector, for example, which is really good at for quantitative analysis. So in that case, you don't get the same kind of structural information, because anything that has carbon is just burned in there and gives you a signal that corresponds to the number of carbons that go through the detector at any given time. But as I said, it is extremely good for quantitative analysis because it decreases the amount of extra work that you have to do if you were to use a mass selective detector. So long story short, what I'm showing here is chromatograms that were obtained with when using various different methods of sample processing and then downstream processing as well. So whole flower, just using a headspray syringe, you can see that the dominant products that are analyzed are monoterpenes. Now, if you use the SPME tool, also with whole flower, then you can see that in the sesquiterpene region, those are metabolites that are slightly bigger than monoterpenes. They contain 15 carbons rather than 10. And thus elute a little later. You can see that there is a substantial increase in the number of sesquiterpenes that we're detecting. Now, even when we use a homogenate, okay, if we still use that headspace syringe, we will have a bias toward analyzing monoterpenes. If we then use the SPME tool again with the flower homogenate, you can't see this, but if you look at the scale, our overall abundance of peak increases tremendously over that you get with the whole flower. And you can imagine doing various different other things. I just wanted to mention one more arrangement here, which is to generate a homogenate, to then extract it with hexane as a solvent, and then do a liquid injection. And as you can see, the spectrum of various different mono and sesqui terpenes is the most diverse. So your coverage using that method is the best. So does that mean that you should always do the method that I've shown you down here? It really depends. For example, if you are someone who develops new chemovars with special aromas, and this is for the fresh flower market, then maybe what you should do is do something that's a bit closer to what your nose actually perceives, because your nose functions more like a headspace syringe, right? You're taking in those aromas directly, and there will be a bias for initially detecting monoterpenes. So if that's your goal, then that would be the method of choice. If you want a very comprehensive analysis, then the liquid injection might be your best choice in that case. And so depending on your application, you might choose which of the combinations of different methods is the most appropriate. So then I wanted to touch on the types of experiments that you would do to ensure that you have consistency. So, you know, when we're asking ourselves which terpenes accumulate in cannabis, then obviously that not only depends on the genetics of your chemovar, it also depends on the environment. So for any study that tries to build on acquiring data at various different levels, you want to make sure that you control for the genetics by environment interactions. And this was work done by Hannah and Zach, who work for Dewey Scientific, that wrote up methods that you can use for mixed light condition growing of cannabis. So this is when you're growing in greenhouses with supplemental lighting. And those are extremely detailed methods, really step-by-step methods that tell you everything you need to know about how you control environmental conditions, how you set up your sensors, how you use various different control systems to adjust pretty much all the parameters that are relevant for a grow, and then also for the overall control of your greenhouse, which is very important as well. And as a last bit of additional data here, I wanted to show you what the pest management schedule could look like. And it will depend on the development stage of the plants. This here is in early flowering, and that will change slightly when you move into flowering. But as you can see, you have a schedule where you're doing certain things at specific times. And that really is the only way you can ensure that you have a consistent grow and you don't run into problems with pests. So then talking a little bit about terpene synthase. So those are the enzymes that are at the core of making terpenes, and that is true not only in cannabis, you know, it's true across species. And it's a topic that is at the core of what my lab does. So the people that were involved in these studies are Shri, who is an assistant research professor in my lab, and has been working on terpene synthase structure function relationships for over a decade. And we collaborated with Ho-Shin Kim and Simone Roger at the Pacific Northwest National Lab because they have one of the fastest supercomputers in the world and can generate simulations at various different levels that have really helped us to better understand how terpene synthesis manage intermediates in the reactions that they catalyze. But before I will talk about this, I just wanted to take one step back and let you know something about the known diversity of terpene synthesis in cannabis. Okay, so when you look at the various different chemovars that people have worked with and then characterize terpene synthesis, there's about 30 genes that people have characterized. They fall into various different classes, but what becomes immediately obvious based on the color code that I used here is that there are some terpene syntheses that are being characterized again and again. So for example, those that generate a lot of beta-myrcene or beta-ocimene or limonene or linalool and so on. So if you add up all the terpenes that are generated by the functionally characterized terpene synthesis, it's not too many. So if your goal is to increase the diversity or maybe even develop radically new aromas, then you would either have to modify what one of the existing terpene synthesis does, or you would have to tap into the genetic diversity in the genus cannabis and then see if there might be something that we have missed because we've focused on such a narrow set of chemovars that people grow commercially. So what's important when you're trying to understand terpene synthesis in the context of what the cannabis genome provides, it's always instructive to look at where those genes are located. And I'm using the CBDRX genome as a reference in this case. So you can see there is a smaller cluster on chromosome 1. There is a larger cluster of terpene synthesis on chromosome 5 in the telomeric region up here. There's another cluster that's larger on chromosome 6, again in the telomeric region. And then there's another smaller cluster on chromosome 9. Now, it appears that a lot of what determines what we find in commercial chemovars is related to those terpene synthesis on chromosome 5. So to give you an idea of how these enzymes work, we're just going to go through a few slides that explain the reaction mechanism. So I'm focusing on monoterpenes here, which are C10 compounds. The precursor in cannabis is primarily geranyl diphosphate, or GPP. And the first thing you do is you abstract this pyrophosphate, and this generates a carbocation. And these carbocations are the hallmark intermediates in the mechanism of different terpene synthesis. And this is important because these are very, very reactive chemical species. In solutions, they would not exist for more than a nanosecond or so, just because they're so reactive and they want to immediately react with a solvent or something else. So what happens in these enzymes, they are able to stabilize these carbocations through interactions with residues in the active site of the enzyme. And what our work established is that it is the differential stabilization of various different intermediates that decides what kind of product you're getting. So moving on, after the geranyl cation, what could happen is that you immediately react with either a catalytic base in the active site. So you would generate a hydrocarbon like ocimene, or you may react with a water molecule that is present in the active site, which would generate geraniol in this case. Another option is if your enzyme is able to stabilize additional carbocations, is that you get the pyrophosphate re-added. However, this is now in a different position. It is here on carbon 3. What that does, it allows this allelic C3 moiety here to rotate, okay? Because now, before you had a rigid double bond, but now you have a single bond, you can move this around. And once you've done that, you could abstract the pyrophosphate again, which gives you a linolyl cation, okay? And again, from there, you could have a deprotonation or a water capture to give you various different end products. So this explains how some of the acyclic, the non-cyclic monoterpenes are formed. But as I mentioned, there's a lot of diversity. So in some cases, starting from the linolyl cation that you've seen before now, you might be generating other intermediates. And those are all carbocations. Now, after that rotation of that bond, you can see that the double bond and that positive charge are much closer together. So what you might get is a cyclic intermediate. And this is called the alpha terpinyl cation. And from there, you could terminate the reaction, or you could form another, or you could have another cyclization to occur, which generates bicyclic products. And in addition to generating mono and bicyclic products from this intermediates, you can go into various different directions where the stereochemistry differs, okay? So you can have one series that generates things like minus terpineol and minus limonene, or you could have a different one that generates terpineol and plus terpineol and plus limonene. So the initial binding of an intermediate also determines the stereochemical course of the reaction. And this is very complicated, so I'm only going to spend maybe a minute on explaining what we did here. We looked at crystal structures of enzymes that make monocyclic products like limonene synthase and those that make bicyclic products like bornyl diphosphate synthase. And we then asked, and those are very complex computational simulations, why does one prefer to generate bicyclic products like the bornyl diphosphate synthase and the other one monocyclic products like the limonene synthase? And what these computational investigations indicated is that for the bornyl diphosphate synthase, this is the free energy profile that you get down here when you assume that you have various different angles between carbons 3, 6, and 7 here, okay? So this is that cyclic ring here, and then you have that side chain. So what you can see is that there is a minimum in the free energy that corresponds to a certain conformation of this intermediate, which we call the axial conformation. So in this case, you're bringing C3 and C7 relatively close, okay? And what that allows is for a new bond to be formed between these two carbons. So the conformation that is energetically most favorable actually then determines the type of end product that you're getting. This is different in the limonene synthase. In that case, if you look at the profile, which is shown on the graph here, the conformation that is energetically most favorable would be one where this side chain is away from that carbon 3, okay? So you've got kind of an extended conformation, and we call this equatorial. So in that case, those carbons 3 and 7 are so far away that it is unlikely that a new bond would be formed. And in fact, what it does, it positions this methyl group down here very close to what we believe to be the catalytic base in the active site. So you prefer a deprotonation as termination reaction rather than the formation of a new bond. So based on that knowledge, what you can do is you can align just the residues that we believe to be relevant for the active site of the enzyme. Okay, so the numbering here up here is from the limonene synthase of spearmint because we have a crystal structure and know a lot about it. And then we're showing different types of cannabis monoterpene synthases here. And those that make monocyclic products, you can see they have a certain pattern of residues that is somewhat consistent. And that is different from the pattern that you find in bicyclic monoterpene synthesis. And we explored that extensively, but I won't have time to talk about this. What I will talk about briefly is how it is decided if these enzymes make purely monocyclic monoterpenes, or I should say not monocyclic, but if they generate primarily monoterpenes, which are C10 products, or if they can accommodate a different type of precursors and actually can also make sesquiterpenes, because we found some terpene synthases that could generate both C10 compounds and C15 compounds. So I'm first going to talk about an enzyme that can only make the C10 compounds, TPS14CT. We tried to dock various different ligands, and as you can see, when you look at the RMSD value, which is a value that tells you how well a predicted structure can be overlaid with that of a reference, and the lower the value in this case, the better. You can see that trans FPP, which is the C15 precursor, is not really accommodated in there. And the reason for that is because we have regions in the active site that we call plasticity regions, that where there's a lot of variation in terpene synthesis. So in this particular one, you can see that the plasticity region 2 and the plasticity region 1 come very close together. They almost seal the active site so that only a carbon 10 precursor can be bound. This is the GPP. However, if you look at enzymes that can generate both monoterpenes and sesquiterpenes, you can see that the two plasticity regions are not as close together, so you can accommodate both the geranyl diphosphate and the C15 farnesyl diphosphate. So this gives us some idea of why some of those terpene synthases are specific for just C10 products, with others are a bit more promiscuous in the binding. And with that, I will come to the conclusions of the topics that we've covered. So I want to highlight again that when you combine genetic and metabolite analyses, then this is very powerful if you're trying to differentiate chemovars, and this is probably something that we should do in the future, where we're going away from wonderful sounding names to something that is more of a scientific annotation. Then I hope I could convince you that we need higher standards for how we identify and quantify metabolites at the analytical level. It is very important to be able to control your environmental conditions if you're trying to support omics studies or genetic studies, breeding studies, whatever it might be. And then something that I would also like to mention is that if you're trying to explore the chemical diversity in the volatile realm, then that would be something that is going to be of critical importance for developing chemovars with novel aromas. So with that, I'll close. I think I already highlighted all the people that contributed substantially to the work. So those are shown in blue here. And with that, I'll close, and I'd be happy to answer any questions or talk about some of your comments. Thank you.
Yeah. Thank you, Dr. Lang. We have time for a few questions. Please submit your questions in the Q&A. I will start with one. I really enjoyed your presentation, and thank you for your insight on terpene analysis. And also, I like the enzymology and the enzyme activity analysis. So my question is, have you also compared the terpene profiles after heating the flowers, right? How does that change? Is it more complicated? Because I think, you may have additional compounds, aldehyde or some other volatiles that may interfere with your analysis.
Yeah, that's a really good point. So when we do our analysis with headspace for headspace composition and also with those BME fibers, we actually move those tubes into what's called an agitator. So there's a chamber that holds various vials and is generally heated. What the heating does is it releases some of the volatiles that might be contained within the glandular trichome. So you're getting a richer profile in the headspace. And the temperature that you're using, I didn't show this here, but the temperature that you're using in there has a tremendous effect on which types of volatiles you're seeing and how much of those. In addition to that, we also tried to look at samples that had essentially been pyrolyzed before, so at much higher temperatures. And you see that the profile of terpenes changes completely. So if you're comparing samples that are that reflect more what you see in the fresh sample compared to what you see in the smoked sample, there are huge differences. And then, as you mentioned, some of the other aroma constituents, I only talked about terpenes, but they're not the only volatiles that are important in cannabis. Small chain esters, alcohols, aldehydes, sulfur-containing volatiles, they all contribute to the overall aroma. And they are much more volatile than the terpenes. So there are additional challenges when you're trying to analyze them. That's a very good point. Thank you.
Great. Oh, I think, wow, we just got a question. Well, thank you for the great presentation. You probably can see the question too. Has your team attempted to create novel terpene synthesis?
Yes, we have. That not with the cannabis terpene synthesis, but we've embarked on extensive mutant generation projects. In some cases, just exchanging individual residues, or in other cases, exchanging entire regions. The goal for those has been primarily to understand which of the residues are important for specific aspects of the catalytic mechanism. But you can imagine that in some of those cases, when you're changing the terpene profile that is made by a certain terpene synthase, you could theoretically do the same in, you could use biotechnological approaches to do that. Now, the challenge with plants currently would be that this would be genetic engineering, and there is a challenge with consumer acceptance of that. There are more recent developments where some aspects of genome editing do not fall under the same kinds of rigor that is required for registering genetically modified organisms. However, this is currently so restrictive that it is hard to see how you could get away with something that is not regulated when you're trying to change the profile of a particular terpene synthase. Oftentimes, you need more than just one residue or two that you need to change. And once you do that, you fall under the very onerous regulation that has been put in place for anything that you call a genetically modified organism. But yes, it is possible, purely technologically.
All right. We're getting close to the hour. I will just make a quick announcement for next month's forum. So our next forum will be on April 13th, also at 11 o'clock in the morning. Dr. Yasmin Hurd, who is the director of the Addiction Institute at Mount Sinai, will speak on translating cannabis from molecule to mind and treatment. I hope you will join us again in April. And let's thank Dr. Lang again for the wonderful presentation, and for everyone, please have a wonderful day. Yeah. Thank you.
Yeah, thank you so much for having me.
Bye.
Bye-bye.